Good morning, students. This is day 2 of the RT-PCR lab. Now, if you recall, yesterday what we did is we took our tube and we amplified, we made some cDNA copies of all of the mRNA that a mouse liver would produce. Right? So we've got our, just to review very quickly, we've got our tube here. And we've got lots of different kinds of mRNA, hundreds and hundreds or thousands of copies of mRNA, all doing different things. They're making all kinds of different enzymes, all kinds of different proteins. We didn't specifically select any mRNA. All we did yesterday is add a whole bunch of tails of T's, so they would bind to these poly A tails of all of the mRNA that's in our test tube, and then we allowed reverse transcriptase, an enzyme, to go backwards along here. It binds to these poly T tails, right? It binds to all of the T's, and then it runs backwards, and it makes a cDNA copy of the RNA. So remember, we made some hybrids. We have some mRNA strands up top, and then our reverse transcriptase made the complementary cDNA strands, which are DNA strands without the introns, right? We have no idea what the introns are. All that's left in this mRNA is simply the exons. So it's not true DNA, it's cDNA, and that's what we've made. All right. But of course, the name of the lab is RT-PCR of GAPDH gene. So we want the GAPDH gene, but up here in our test tube, we have the GAPDH gene, but we've also got hundreds and hundreds and thousands of other mRNA, right? And that's not what we want. We want to get rid of all of the other mRNA, and we want to specifically select for just the GAPDH gene. So that's what we're going to do today. Day 2 is selecting specifically for the GAPDH gene, and we are not going to amplify any other segment of DNA that doesn't have GAPDH. So here's what we're going to do. We're going to add a little bit of our master mix at our lab station. So what we do is we make up a master mix that's got all of the stuff in it. Buffers, primers, enzymes, nucleotides, everything that we will need for a PCR reaction, we put all in one tube. It just makes it easier to aliquot that out into the individual tube, and so we don't leave anything out. All right. The really important thing that is in this tube, though, is right here. This is the primers, right? The primers that are in this tube, what their job is they will specifically, very specifically, select only for the GAPDH gene. Right, these primers will not bind to any other gene that is in our cDNA strands if it is not specifically GAPDH. And we know how it does that, right? We can come over here and let me go back to green real quick so I can show you this, right? So here's the cDNA strand that we've made, cDNA. And along the cDNA, we've got a very specific sequence of nucleotides. Could be A, C, T, T, A, C. It doesn't matter what it is, right? But it's very specific. And it comes just before what we want, right? So this section right here could be GAPDH. And just before that, we've got this very specific sequence of nucleotides, and we can take advantage of that. We can actually call a company and give them this sequence of nucleotides right here, and say that we'd like a primer that's going to bind to that. And then what it'll do is it'll run along the cDNA that we've made, and it will amplify the GAPDH gene that we're interested in. All right, so if you remember from our transcription and translation talks, we need the complementary strand to this. So our primer that we have in the tube is going to specifically be a sequence of nucleotides. That's all it is. It's a short sequence, usually 20 to 30 base pairs. In this case, I didn't draw 20 to 30 just to save some space. Right? So we'll draw the complementary. G-A-A-T. Remember, it's DNA, so we're using T's instead of U's. Right? So this here is the primer that we've purchased, and it will bind only to the sequence just before the GAPDH gene. And then what we're going to do, I'll go back over here. The second most important thing that's in our tube is this right here, enzymes. Right, specifically our Taq polymerase enzyme. Right? If you should remember this word, polymerase, this should sound very familiar, right? DNA or RNA polymerase. And in this case, Taq is simply a generic DNA polymerase. So this will make DNA. All right, so this Taq polymerase, let's see, I'll use a big color, I'll use pink for it. It will bind onto our primer, and then it will go down the line, and it will insert nucleotides. It will make a new DNA strand, right, using the primer and copying this GAPDH section. It will be the complementary strand. All right. We also have a primer that goes the other way, and then we'll make a copy going this way. That's a little bit more advanced. We can talk about that at a later point. All right. But today, all we're trying to do is we're trying to separate out everything that's not GAPDH. So let's say this is our GAPDH section right here of mRNA that we want. We're trying to get rid of everything else that doesn't code for GAPDH, and that's what today is. All right. So we're going to add all of this stuff to our PCR tube. Notice we're going to save our single-strand cDNA. That's our tube from yesterday. So we're going to add a little bit of our tube from yesterday, the single-strand cDNA, and the PCR master mix. That's going to be the tube that we're going to use today. We're going to store the one from yesterday in the freezer. And then we're going to run a PCR cycle. All right, and then we're actually going to run a couple more. Notice we run 34 cycles to amplify the GAP gene. So what happens is every time we run a new cycle, this Taq polymerase will run down here and make another copy of DNA. Right, but now we've got 2 strands that we can copy. All right, and so actually the Taq polymerase will jump on 1, jump on 2. It'll now make 2 copies. So now we have 1, 2, 3, 4 copies of our gene sequence, and Taq polymerase can use all 4 of those as a template, right? So do you see that very, very soon we will have exponential growth going on. We will exponentially copy DNA. So after a few cycles, after 6, 7, 8 cycles, we will have millions and millions of copies of our DNA strand, right? All of this original stuff, though, this is very important, all of this original stuff is still in the test tube. We do not destroy it. It does not go away. The only thing is it will be in such a small amount, there might only be a strand or 2, that when we run it on a gel on day 3, there's no way that it's going to show up. The gel is not sensitive enough to make that visible. What will show up, though, is these millions and millions and millions of copies of just the GAPDH gene, and that's what we're going for. And that will be day 3. So day 2 is simply adding a few things to the tube, and then the PCR machine does the hard work of 34 cycles, and then it will hold our tubes. It will stay at 8 degrees Celsius. So nice and cool, just so nothing will happen to it. It can stay that way for a really, really, really long time until we go to day 3, and we check our results on a gel. We'll run gel electrophoresis on day 3, and that will be the next video. Thank you.